Looks like people just assume that since there was a quiz last time, there's not going to be a quiz today, so nobody came to class. Either that or just all sleepy. All right, so last time we talked about more some of the gene regulation. We talked about this, just like the general concepts of how gene regulation worked, and then we talked about the two main cases that we talked about were the lactose operon and the tryptophan operon. So here's some review questions. So can anybody tell me the answer for the first one? Yep, good. Oops. And then where does allolactose actually come from? I am hearing partial answers. I can't tell who's saying them though. Yeah. Yep. So Laxi, it basically rearranges lactose. It can't fully cleave it, so at that point, so it rearranges lactose to allolactose. All right. So today we're going to talk about more types of regulation. So we're going to move on from the protein regulators a little bit and then talk about other ways to control them using sigma factors, small RNAs, and then we'll talk about some of the bigger kind of combination of these different regulation type things. All right, so one other way that you can control transcription is through different sigma factors. And so if you remember from the chapter previous, we talked about that sigma factors recognize different promoters, right? So that is just one simple way of regulation, is just having a different promoter that will be recognized by a completely different sigma factor. RPOD, so sigma 70, is the one that does all the general housekeeping genes. So those are the ones that are most common, the ones that are on all the time. But the other sigma factors, so some of these other guys here, are for other environmental situations, and so they're only around when the cell is experiencing a certain condition in the environment. So there are different ways that the cells actually control the process of making the sigma factors themselves. So we'll go through a couple of examples. So the first one, it controls the degradation of the sigma factor. So RPOS, which is the one that controls the genes that are needed in stationary phase. So that one, the cell actually just controls how fast it's degraded. And so during exponential growth, RPOS gets degraded really quickly. And then during exponential growth, everything is slowing down. I mean, during stationary, everything is slowing down, right? The cell's not actually growing. And so that's when RPOS doesn't get degraded, and it can actually then go and turn on the stationary phase genes. The other way that sigma factors are controlled is with an anti-sigma factor protein. So these anti-sigma factors inhibit the sigma factor from actually binding to the polymerase. So remember, the sigma factor is just has a spot in the RNAP, and that whole thing binds, and then that all is what binds to your DNA. So an example of this is in flagella synthesis, and so FGLM is the anti-sigma factor, and so this is secreted from the cell when flagella are necessary. And so so we have the anti-sigma factor that will actually block this when when flagella are not necessary. So this is what this is going to be blocking the sigma factor from binding to the RNA polymerase when you don't actually need that synthesis. And then there's anti-sigma factors. So again, we're adding more things onto the regulation. These bind up the anti-sigma factors more, and so the binding between anti-anti and anti-sigma factor is more tight than just binding of the anti-sigma factor to the sigma factor. So it's going to want to bind the anti-sigma factor, and that will essentially decrease that concentration and free up the concentrations for the sigma factors themselves. The final example that we have are is for the RPOH. So these are this is the sigma factor that controls the heat shock genes. So it uses an RNA thermometer to control the translation of this RPOH sigma factor. And so it's called an RNA thermometer because it is essentially sensing the temperature. Yes?
Sorry, I had a question. For the anti-sigma factor, you
yes.
said that it blocks the factor?
Yeah, so the anti-sigma factor inhibits sigma factor from binding to RNAP.
Oh, okay.
Yeah, so it blocks. It's almost as if it's just like preventing it. It's like blocking it from binding to the.
Sorry, what was RPS?
Ribosome binding site. Okay, so like I said, the thermometers, they're called RNA thermometers because they're essentially detecting the temperature that the cell is in. And so it does so basically by detecting because it changes the bonding and of the RNA structure itself. So at 30C, this is essentially considered a low temperature for the cell, or the normal growth temperature for the cell. The RNA structure will form that will block the RBS. So just for example, and this is not necessarily what everything is going to look like, but let's just say it's going to form a hairpin, right? And let's say that the RBS is here. And so now if that RBS is all bound up here, it's not going to be accessible for the ribosome. But at 42, this hairpin now is going to melt because the temperature is higher. And so this will become straight lined like this, and you'll get your RBS is accessible. And so now you can get translation of that gene. Does that make sense? Yeah.
For the anti-anti,
yes.
So is it blocking the anti-sigma factor from binding, or is
yes.
it fully
So this will bind to this, and so it essentially it essentially sequesters that away, and then so then the anti cannot bind the hip. Okay. All right. So the RNA thermometers were kind of a preview into one of the types of RNA regulation that we can have. But there is this whole class of RNAs called small RNAs that we denote with the siRNAs. So these are typically post-transcriptional control. So that's just, depending on so that just means that it can control, mostly it usually controls translation, but it also can control how fast the mRNA gets degraded. So many, but not all, require this chaperone called HFQ, which is a hexameric protein. So HFQ has two purposes. It'll actually bind to the siRNA and stabilize it, but it also helps bring the siRNA in contact with the mRNA that it's regulating. One of the key things to note about these siRNAs is that they can actually regulate multiple genes. So the siRNA is just one standard sequence, but it can bind to multiple genes. So it doesn't actually have to bind exact complementary. So it doesn't, if it's, say the siRNA is 80 nucleotides long, not all 80 nucleotides have to bind to the mRNA for you to get regulation. So as long as there's enough binding to block or activate, as we'll see on the next couple of slides, that's all that's required. So you can actually have one siRNA that kind of matches the sequence on this one, but then there's enough of it that matches a sequence on a completely different gene, and it'll be able to regulate both. So here's just a list of examples of some of the different siRNAs. I don't expect you to know what they do, what each one does. I do expect you to know what I'm going to go through next is the different styles of regulation that they do. I believe that in E. coli there are 30 of these known siRNAs, and those are only the ones that actually require that HFQ. And so there are many, many more of these siRNAs in different cells. Okay, so the first thing that it can regulate is translation. And so it can do both inhibit translation, so like stop block translation, or it can also activate translation. So in the case of inhibition, so you'll end up having the sRNA, literally whatever the base pairing, will bind to the RBS and block it, so you can't get, the ribosome can't access the RBS. In other cases, what can happen is you can actually get the sRNA to bind someplace else on the mRNA. So in this case, what we have here, the mRNA itself is folding and and blocking the RBS itself. So the mRNA, the one single strand, is actually folding upon itself and blocking the RBS. But what you can have is an sRNA come in and actually bind to this part of the mRNA, and now this essentially would prefer to bind to the sRNA, and that opens up the RBS here, so that activates translation. We can also have sRNAs that control degradation. So degradation of RNAs are done by proteins called RNases, and RNases will recognize different sequences or different structures in RNA structures, and so you can actually get sRNAs that will bind this, and in this case, this RNase is going to recognize this double-stranded RNA, and it's going to target that as it's going to start chewing that up. In other cases, though, you can have, so let's say this RNase actually recognizes this single strand, and maybe it has the proper sequence for this to chew up, but now the sRNA is going to bind here, and it's going to actually stabilize that. And so normally what that means is the mRNA, the thing that's that needs to be translated or whatnot, is downstream from here, and so this is now just preventing the whole thing from degrading. And so how do we relate degradation to actual translation? So what happens if this, in this case, do you expect translation to be, expression of this protein to be higher or lower? Lower. Good. And then in the case of this case, of the, when it's preventing degradation?
Higher.
Good. All right. So another class of antisense RNA, of regulatory RNAs, are called antisense RNAs. So usually they are called cis antisense, meaning they're transcribed from the same gene that it's going to be controlling. So it's transcribed from the non-template strand of the DNA. So because it is actually transcribed from the non-template strand, it's complementary to the mRNA itself. So what can happen is that it's going to be perfect match, right? So it's going to perfectly bind to the mRNA sequence. And this can, there are many different examples of these. I'm not going to go into them. Some of the mechanisms get a little bit complicated. But so the key thing about these antisense RNAs, it's going to only regulate the gene that it was essentially being transcribed from, right? So this is contrary to the sRNAs that we just talked about, where I said that one sRNA can go and regulate multiple genes. These antisense RNAs, because of the specificity, because of where it's coming from, will only regulate that gene that it's that gene that it was encoded from. So, but you can, again, these antisense RNAs out there can control transcription, translation, and mRNA degradation. One more class of RNAs is called riboswitches. So RNAs can also bind metabolites. And so it's not only the proteins that can bind these small molecules. RNAs can actually bind them within their sequences. So they bind things like amino acids and vitamins and even some metals. So again, these riboswitches are found in the 5' untranslated region of the gene. So the untranslated region is basically anything before the start codon. And so what happens is that you can get, when you get the ligand binding to the riboswitch, binding to this RNA, usually what happens is there's going to be some kind of conformational change downstream of where that's been binding. And those conformational changes can, create these different stem loop structures. But what they can do is both translation control and transcription control. So you can have the cases where you are, again, either blocking or giving access to the ribosome binding site. And then in the case of transcription, so again here, we've talked about this a few times now, is that intrinsic terminator stem loop structure. So the thing that your picture is missing, so you would imagine that there would be U's down here. And so binding of the ligand to the riboswitch can lead to a conformational change that will then lead to the formation of this terminator hairpin, and so you get stop transcription there. And there are cases of both of these that will either activate or repress both transcription and translation. All right, so that's kind of the individual like ways of regulation. Now we're going to talk about some of like how the cell kind of puts all these things together. There's all the different components and makes these integrated control circuits. So one of the classic examples that we learn about is chemotaxis. And so we'll just recap a little bit of what flagellar movement is. And so remember that again, so there's also genes that regulate when the cell is making the flagella, but once it's made, they will move in the counterclockwise direction, and so all the flagella sweep behind the cell and it propels the organism forward. So that's considered a run. So counterclockwise considers a run, and it's always trying to basically find food or move away from something that's toxic to it. The example we're going to walk through today is about when it's moving towards an attractant and how it's finding its better environment with more food. But then the flagella can also move clockwise, so it moves in the other direction. Essentially, that will disrupt the bundle and the bacteria will tumble. And so essentially the bacteria is moving in one direction and then it's going to go the other way, and that's going to cause it to tumble, and then it'll kind of tumble in a random direction. And then it's going to keep going counterclockwise again, and then the bacteria will move forward in whatever direction it got situated to. And so it's going to keep doing that until it kind of senses whether it's in a better spot than it was before. And essentially the number of tumbles are suppressed as it moves from a lower to a higher concentration of attractant, right? So as it's going, it's sensing that it's getting closer and closer or getting more and more food in the direction that it is moving. It doesn't want to like actually move in another direction, so it's going to slow the number of times that it actually tumbles. All right, so how does it do this? So flagella, again, the natural movement is counterclockwise. In order for it to actually tumble, what the cell does is it phosphorylates the protein KY. So this thing here gets phosphorylated. Then KY actually interacts with the rotor. So if this is the rotor, it actually interacts with this rotor, and it causes the rotor to turn the other way. So how does QI actually get phosphorylated? That's by this KA, which is a protein kinase. So these guys here are the ones that are going to phosphorylate the KY. But then you also have KZ, which is going to continually dephosphorylate. So once this comes here and it flips this switch, it's going to get dephosphorylated and it can get used back again in the system. But the tumbles aren't continuous, so the cells can't go continuously clockwise. And so it's just going to flip it for a little while, have it change its direction, and then it's going to go back to moving the other way. And again, so increased attractant decreases the KA activity. So the key is the activity for which KA is going to phosphorylate KY, and that's how it determines how often you get the clockwise movement. Back, you see how this here was in this conformation here when there's no attractant, but yes.
So if you phosphorylate it, that just means it's going to rotate clockwise?
Yeah, so you phosphorylate it and then just essentially think of it as it like bumps into the rotor and it causes it to go the other way for a little while. Okay, so how does the cell know that it's actually reached the attractant? Because it wants to like keep moving towards an attractant, but then once it does that, it wants to actually stop it from going the wrong direction, right? So what it uses are these methyl-accepting chemotaxis proteins, this MCP, so that's this guy here. So the periplasmic domain, so this side, it binds to the attractant. So it's going to recognize something that the food that it wants or the chemicals that it wants to move towards, and that's going to bind to the MCP. The cytoplasmic domains of the MCP is what actually binds to this key A through key W. So it takes two proteins here to actually bind to this. But when this is bound, you see how just remember what this looks like. When the attractant is bound, this changes conformation, and now this separated key A cannot actually phosphorylate key Y. So binding of the attractant to the MCP causes this conformational change that inhibits key A, and so now you can't get the phosphorylation, and so therefore you get less tumbling. So that's essentially just once, right? Like when the first time it's bound, but sometimes it might see the attractant, but then there might be places that's higher in concentration, so it still maybe wants to move more in that direction. So how does it know if the concentrations of attractant are actually increasing? So that conformational change in MCP also subjects the cytoplasmic side of the MCP to methylation by key R methylase. So this part here gets methylated here. And what essentially happens is that reactivates the whole thing. So the attractant will come off once this is methylated. But now that this is methylated, this part here becomes more sensitive to the concentration of the attractant. And so now in order for the attractant to actually bind here, it has to be in higher concentrations in the cell, in the surroundings of the cell. All right, so again, like I said, so key A is reactivated, but it's desensitized, so that has to be higher. So now it's still going to be moving, but then in order to inhibit key A again to tumble, it's gonna need a higher concentration. So when the cell, the cell is it does all these movements randomly, right? So if it gets it wrong and it moves away from the attractant, so the methylation can be reset by this key EB, this phosphorylated of key EB. So key A also phosphorylates key B, but so this will actually remove this phosphorylation and it resets the sensitivity. So now it can, if it was in a lower concentration, now it can go back to try and find that original spot that it was in. You got a question?
So initially you said that it makes it more sensitive or less sensitive?
So it requires it, so it desensitizes, meaning that it requires higher concentration of the attractant to bind to trigger it. All right, so there is a video of this that I posted on Canvas that I encourage you to watch, but I will just kind of, again, like overview the big points. So again, so naturally the flagella are moving counterclockwise, right? So it needs a signal to tell it that it's going to go clockwise, and so for that, the actual signal is the KYP, and so the phosphorylated version of KY. And so I'm just going to draw and like kind of track the different components of the system. And so we have one of the key components is this KY, right? And so this is going to go back and forth between the phosphorylated version. And so to be phosphorylated, it's going to need key A. But you also have to maintain the concentrations in the system. You have key Z that dephosphorylates this, right? But this is the thing that flips the rotor clockwise. So this is the thing that causes the tumbles. Okay, so then you want the cell to be able to figure out, is it going into a region that has more attractant or less attractant, and did its tumble take it into a place that it wants to be? And so to do that, we have the MCPs. And so the MCP plus the attractant. So this thing essentially turns off key A. So this thing gets turned off. So this part here gets turned off. And then So if this is turned off, then you'll get less of the clockwise movements, right? But we don't want this to just stay like this forever. So we want to be able to say, just because it found one molecule of attractant doesn't mean that it couldn't find more. There might be a place that has better spot. So we have how it regulates that is it goes back and forth between the methylated version of this MCP. And so we have key R, which is the one that methylates it, and then the phosphorylated version of key B that demethylizes it. So this one is essentially this, when it's in this form, it desensitizes the MCP, and then this one here, this reaction here happens so when the cell moves away from the attractant. So it needs a higher concentration of the attractant in the cell for it to bind to that top portion of the MCP here. All right. So there are still other types of regulation. One of them is called quorum sensing, which we've kind of brought up a few times in other contexts. But quorum sensing is essentially a sensing of these chemical molecules, which triggers some kind of behavior. And so it was discovered in the studies of this Vibrio fischeri, this marine microbe that colonizes a squid. So this actually is bioluminescent, but only when it reaches high enough cell densities. So when there are enough of them around, then it causes this luminescence, and in this case, inside the squid. And so how does that actually work, and how does that actually turn on the genes? So induction of the quorum sensing gene, so again, it requires accumulation of this secreted small molecule. So it's called an autoinducer. And so again, it secretes this molecule, and then when it gets high enough concentration, eventually that will come back and turn something on. In the case of this original example, the autoinducer is called homoserine lactone. But we did talk about this in other contexts. Remember how we talked about the competence factor, the peptides for the gram-positive, that's how it turned on whether it was competent for transformation. So it had those peptides that it secreted, and then that was how it knew if there were enough cells around that it would start letting in DNA. This one works the same way. So the more cells that are in kind of this discrete place, the faster the level of the autoinducer will accumulate. Once the autoinducer is at a high enough concentration, it will actually re-enter the cell. And in this case, it will bind this regulatory molecule called LuxR. So the LuxR autoinducer activates transcription of this luciferase gene. So that's the thing that, luciferase is the thing that leads to the bioluminescence. So again, you have the autoinducer leaving, but then once it hits high enough concentration outside of the cell, it'll come back in. So this is just one example. So that was the first example that discovered it, but there are different examples of these quorum sensing systems in different species. Again, this is not something that I expect you to know what all of these species are, but just know that this kind of communication, right? So in this case, it's communicating whether there's more of your friends around, but other times it can actually use to communicate between cells, so also between species, so it can communicate with other species with some of these systems. All right, so the last kind of gene regulatory example that we'll go through is we're going to return to phage lambda and the regulation of lysis versus lysogeny. So we had talked about that the cells there are some viruses that can do both lysogeny and lysis, right? And then we just kind of skipped over how does it decide which one it's going to do, right? So whether it's going to be in the lytic cycle or the lysogenic cycle. So what actually determines this is concentration of two different proteins. So the CRO protein is the one that induces lysis. So small amounts of this CRO protein will activate the lytic cycle. This is versus the concentration of the C2 protein. So this is the one that will have the phage enter the lysogenic cycle. So C2, and we'll get into this, actually induces expression of C1, which is known as the lambda repressor. So that is the one that blocks expression, kind of like the other repressors that we've been talking about already. So this actually blocks expression of CRO and the lytic proteins along with it. So which one gets expressed? So that kind of depends on many things. It depends on the environmental factors of that the cell is experiencing, the presence of other phage. So just some examples. So high nutrients will actually cause, so there's a protease within the cell that will actually cleave the C2. And so if C2 is cleaved, then you're going to leave the CRO protein around. That will induce the lytic cycle. In low nutrients, C2 concentration actually remains high, and so this continues to block CRO, and so you get lysogeny. Another example, so during DNA damage, so remember when we were talking about DNA damage, remember this REC-A protein that we kept talking about? So REC-A actually cleaves C1 during DNA damage, and so that will essentially relieve this repression and then lead to lysis. We're going to go into all of this right now, so yeah.
So it says C1 block expression of CRO, right? So there's low amounts of CRO still?
So there, essentially it's going to block it, so there's none. Yeah, we'll get to it, yeah. All right, so this is what the DNA for just this part of the regulation looks like, and this is how your book represents it, and I found it a little bit confusing, and so I have changed it and color-coded it so that you know which promoters are controlling which genes. And so the promoter colors are matching the genes that it's going to actually be, that are actually transcribing. So it gets really confusing because this green one here, it's over here, but it's not actually controlling CRO, it's only controlling C1. So this color coding will hopefully help you remember that. Okay, so now let's just imagine the phage enters the cell. What happens is it's going to start with transcription from both PL and PR. So as soon as it enters the cell, you know, it busts open, gets its genome in there, that's what's going to happen. It's going to start transcription from both of these. And these control most of the phage structural genes, and including the lytic genes. So the decision between lysis versus lysogeny essentially just depends on which regulation happens first. So it's kind of a race to see which happens first. So you can see here, so this is going to start being made, right? And this is going to make both CRO and C2. And so they're coming from the same promoter. They're going to both get made, so which one's going to win? So in the case of lysis, CRO wins. So remember that it's not just there are other things that can be in the cell that can be helping this out, because C2 can be cleaved by a host protease, right? So that will essentially decrease this concentration, and so the concentration of CRO will win the race. And so what happens in this case, so CRO is concentration dependent essentially, and so low concentrations of CRO, initially it will block the C1 production at PRM. So you'll get CRO protein, that's what the circles mean. It's going to block production from here, and so you're not going to get C1, and so that's it. It's not going. There's no competition for it to stop preventing lysis. At high enough concentrations, CRO can also block PR and PL. So when it gets to high, so this is going to keep getting made. When this reaches a high enough concentration, it'll essentially block itself and PL. This just kind of, if you remember just how the life cycle of the phage works, there are different like early genes versus late stage genes. And so once it's done with the early genes, CRO is high enough, it can repress those and just kind of move on to the late stage genes. All right, so in the case of lysogeny, obviously C2 is going to win. So C2 is an activator. So C2 activates activates the promoters to PRE, so it activates this guy, but it also activates something that's not on this picture, so it activates this pint, and so this promoter is what actually controls has the enzymes for integration, right? So that's part of lysogeny, right? It has to be integrated into the host genome, and so right, the genes here is only the genes for this switch, but there is a promoter downstream somewhere that's, if C2 is around, it's going to start making the genes that require integration. Okay, so again, C1 is a repressor. So C1 blocks PL and PR. So C1's made, it blocks the promoters here. But actually binding at this OR, if you remember what it said underneath there, it was OR. So actually binding here also activates this promoter here. So it kind of does both. It blocks the PR, but it activates PRM. And so it's essentially making more of itself. And so it maintains its own expression in this case. Fine, it's just sitting here. This is blocking. How is that ever going to switch to lysis? Okay, so once it's in lysogeny, then, right, so you're like, okay. So there can be, sometimes there are just random fluctuations in C1. And so essentially, the concentration of this will decrease low enough that this will actually release from the operator site. And that once this moves, it starts producing the CRO protein, and very little CRO protein is actually needed to switch to lysis. But remember, again, C1 can also be cleaved by REC-A, so during DNA damage, these will go away, and then you'll get switched to the lytic stage. Does that make sense? Okay, so now I'm just going to talk so your book also talks about this example of an application of the CRO C1 switch. I was actually pretty excited that your book actually talks about this, and I was actually pretty surprised because this is relatively new research. Dr. Pam Silver is a synthetic biology researcher at Harvard. And actually, because of this research, I almost did my postdoc in her lab. I was very close to deciding. It was between this one and the lab that I actually did my postdoc in. But it was based because of this research article that your book talks about. So what they did is they took this CRO C1 switch and they simplified. So they removed some of the genes and they made it simplified. And so they made it induction deficient, meaning so you couldn't, there was no C2, so you couldn't induce C1 production. And then they used a strain, they used a modified E. coli strain that had no REC-A cleavage. So you essentially couldn't chew up the C1 either. Then they also, what they ended up doing, so you see the genes here, but the rest of this still looks the same, right? There's still PRM, the PR, the PL, and then it's still regulating CRO, and this one is still regulating C1. But what they did, instead of having the C2 down here, they actually fused CRO to LAC-Z. So we talked about LAC-Z in the context of the lactose operon, right? And how it's going to cleave lactose into glucose and galactose, or it's just going to rearrange it to allolactose, right? But LAC-Z can actually cleave other chemical molecules, and these chemicals lead to color changes. So one example, there are a couple of these chemicals that LAC-Z can actually cleave. So one example is XGAL. And so XGAL, when you cleave it, it changes color to blue. And so what you do is you would actually put XGAL on the plates. So you guys have been growing bacteria on plates in lab, right? So you would actually put XGAL in the media, and if your cells are producing LAC-Z, your colonies will turn blue. If your cells are not producing LAC-Z, your colonies will stay white. So if you were to Google this, you would often see like a blue-white screen. That's what this is also called. Okay, some of the other modifications is that they use this modified E. coli strain that automatically sets the switch to the C1 state. So there's no choice, right? They integrated this switch into the genome, and that they modified the strain so that C1 is the start. It's always starting in the C1 state. So C1 is always blocking this, and it's always making more C1 at the beginning. So if you were to just kind of take these cells and grow them on a plate, you would get white colonies. So initially it was in the C1 state, so everything was blocked. All right, so then they added a second DNA element, which they called the trigger element. And so this one uses the tetracycline promoter system. So this is another one of those protein regulator systems, just like how we talked about the LAC operon and the trip operon. This is just another one of those kinds of natural regulation systems. So in this case, it's a repressor. So the TET repressor blocks the promoter PTET until tetracycline is added. So tetracycline, in this case, the ATC is the inducer. It removes TET-R. So the way that they set it up is that you have TET-R here. This is the repressor protein. Your TET promoter is controlling expression of CRO. And so when ATC is present, so when tetracycline is present, it's going to bind this, and this is going to remove, and you're going to get CRO expression. So in its natural context, this tetracycline repressor, this promoter repressor system normally controls the expression of tetracycline resistance genes. And so when the cell sees tetracycline, and then it's going to turn on the production of these resistance genes so that it can survive in its presence. But now, all right, so what happens now? So we have these E. coli, we have this switch integrated into its genome. We also have the trigger element in there, right? So initially it had now we add this, and then when you add ATC to it, so now CRO is being made from here, and this is going to actually block the promoter that's making the C1, and so eventually your concentration of C1 is going to decrease. Remember, because it's in a cell now that you can't get induced. So this is going to decrease, and when the concentration is low enough, these are going to fall off, and you're going to make the CRO that's attached to the LAC-Z. And so you're going to get the LAC-Z, and then your colonies will be blue. So then what they did is they took it one step further. They took this E. coli carrying their circuit, they put it into mice. So they fed it to the mice. It's like slightly stronger than feeding them to the mice, but they forced them into the mice. They let the mice go for a few days and let the E. coli essentially colonize in their gut. Then they feed it some ATC. So just a short, so here, this triggering here. So for this short time point, they give it some ATC. And then they essentially, for the next several days, they collect the fecal matter from the mice, and then they grow the bacteria, and they look for blue colonies on the excal plates. And so we have mice that didn't get ATC, and then you have the mice that did get the ATC. And so essentially what happens here is the switch flips. In the case when it does see ATC, it flips, and it essentially flips permanently, right? And now it's considered as this memory element. So now, remember the mice saw the ATC here, but it's not seeing ATC here anymore as the days go by. And so now it has actually remembered that it's seen this antibiotic, and you can then track it when it's coming out in the feces of the mice. So essentially this now, you're creating these cells that have all these circuits in them that can actually just kind of check box and record whether they've seen different situations in the mouse gut. And if you can imagine, like that can eventually be translated into humans, right? That you can now have these kind of bacteria that are telling you what happened inside of you, and then that can hopefully one day maybe inform some treatments, or just kind of tell you what's been going on in your system. So I don't know, I thought this was really cool. This is essentially the research that got me excited about the microbiome and things like that. All right, that's it for today. So next time we'll start chapter 13, which is energetics and catabolism.